Uh, Hello, welcome to this session. The first speaker for the day for the session is Dr. Collins Assisi from IISER Pune, who has faced the problems of other theoretical computational science who is trying to interface with biologists, right?
Sorry, I didn't hear that. I'm just too nervous, so I can't hear stuff when I'm nervous. What did you just say? Did you say something about? No, All right. Thanks, Joby. If I vaguely know, you said that I'm a theorist trying to who has experienced. Okay, good. Because I've heard worse. Yeah, all right. Recovering physicist is one. So, all right. So first, let me thank the organizers for the opportunity to talk here. It's wonderful to be in ICTS. Thank you, Neelima. Thank you, Ram and Amit, who's not here for the invitation. So I'm going to talk to you about spatial representations in the brain. And these are representations that we see in a part of the brain known as the hippocampal formation, which has cells that respond in ways that depend on very often the location of the animal, right? But before I go there, I should tell you that the perspective I'm going to take is perhaps not controversial, but certainly a little bit unusual. The argument is, well, before we go there, let me acknowledge, because I typically never get to the end of my talk. So I put my acknowledgement slides at the start. So the work that I'll be talking about is done with Inayat, who's a graduate student in my lab. And some of it was also done when Arun was a graduate student in the lab. And we've had lots of discussions with the other people listed out here, and thank the funding agencies that have over the years supported our work. All right, so the general idea is that brains have evolved to move, to let an animal move, be it moving in space, be it moving muscles that help you sing a song, what have you, right? So a great deal of the way I think about computational neuroscience in general, has to do with the primacy of movement. And so I put this quote by Daniel Dennett, which says that when a sea squirt wanders through the sea and eventually finds a rock, a home to stick on to, it starts off, it has this sort of a ganglion of a brain that it uses to navigate the world. And once it goes and latches on to a particular location, it doesn't need its brain anymore. And so it just consumes its brain. And I've been told that it's rather like getting tenure. But nevertheless, so, much of the ideas that I'll try to talk about have to do with the primacy of movement, or at least that's the germ of the idea out here, even though I'll be talking about systems that seem to be quite far from systems in movement, right? And let's start off by taking a few lessons from this 19th century automaton. This particular automaton is in the Franklin Museum in Philadelphia, all right? And it's got an elaborate series of gears and cams and so on that help it move this rather nicely articulated hand and make these lovely, elaborate patterns, right? So you'll see this image or this image and so on. All of these are produced by this automaton. It's one of the most spectacular kinds of automata that you will see. It's from, I think, the mid-19th century. So somebody dropped this thing off and people were trying to figure out what it does for the longest time, and they didn't know. But someone put a pencil in its hand and then cranked it up and eventually got it to work. And it also wrote out the name of the maker Maillard, right? So it makes a series of 7 images, some calligraphic text like that. There's a poem that it can write and so on. Now, you'll see out here that even, in fact, let me get to the next. So there you see, there's one of the images that it's making. It's completely mechanical, and I'll show you what the heart of this automaton is, right? So at the core of this automaton, what you will see is a bunch of discs like this, right? And I don't remember, it's a few hundred discs at least. And each of these discs has tiny indentations on it. And all of these discs, they drive basically some sort of a lever to go up and down in the right phase, at the right time. And it can generate all of these images. Now, what's really interesting, and I think Srinivas from yesterday would agree with me, is that this is really a bank of oscillators, right? It's a bank of oscillators. It has certain frequencies that it can generate, and there are certain phases that it'll run through. But using something like this, using a bunch of oscillators, this particular automaton can generate over the time scale that we saw seemingly non-oscillatory patterns, right? Now, what does this have? And you know, it's an interesting way to think about maybe non-periodic, perhaps ballistic movements can even be generated using oscillators, using periodic elements, right? Just like Fourier would have suggested, I would guess. So, of course, engineers have been dealing with things like that. So if you wanted to move this pin up and down, you can have various shapes that you use, and you can see that you get very different kinds of movement, perhaps a 1 to 2 kind of a rhythm out here, and so on, right? So you can generate all of these elaborate movements, mechanical movements using extremely simple oscillators with the right frequencies and the right phases that are built into them, right? So the idea out here is that, of course, your brain is replete with oscillators. So if you look at a very simple layer 5 pyramidal neuron, you could give it, you could inject currents into the neuron, and that would lead it to spike. And depending on how much currents you inject into it, you will get different frequencies of firing, right? So here's a single neuron that acts as an oscillator. If you look at networks of neurons, and this is this particular network of this particular pair of reciprocally inhibitory neurons is what's known as a half-center oscillator. And you will find this in all the circuits, most of the circuits that are involved in movement. So what's happening out here is that the red guy out there fires a bunch of spikes. You have a green guy out there, and when the red guy is spiking, it inhibits the activity of the green neuron. And through some mechanism, perhaps some kind of adaptation, you know, there are many different ways that you can do this. The green suddenly flips over, and when it flips over, it sort of kicks the red down into suppression, right? So you have this alternating pattern of activity, which seems to be a key motif that you see in movement in general, right? And this is this kind of an oscillator is something that you perhaps find in the lobster gut, or even in the spinal cord of various animals. Here's another example where each of these, so in this particular case, the oscillators can either be silent or they can fire a set of spikes, right? Just a single frequency. And by connecting them together via inhibition, you have the switching kind of behavior. Here's an example of oscillators that are autonomous. So they would fire on their own without, so they generate these bursts of activity. And when you connect them, it's the phases of these bursts that can change, right? So there are many ways in which you can come up with these oscillators, either with single neurons or with networks of neurons. So what does this buy you in terms of movement, right? So here's an example of one of the small motor circuits that you will see. Here you have 3 neurons, and the interaction between these 3 groups of neurons is largely antagonistic, right? And because of these antagonistic interactions, you will see that the phase of the oscillation that each of them generate is splayed, right? So you have the blue oscillator firing, the green oscillator firing, and this one firing, and they're all firing at different phases. So the connectivity gives you a patterning that can then be harnessed to have movement, right? These are the patterns that are harnessed to drive muscles that in turn move animals around. So here's the kind of output that you'd see from neural oscillators, right? So this is a jellyfish, and you have this net of neurons, and you have traveling waves of activity that go through this net of neurons, and that's what's giving rise to this undulating oscillation that you see out here. If you look at the gait of horses, you will see that there are different phase relationships associated with the movement of each of these limbs, and all of these are driven by motor neurons. So there are muscles that are driven by motor neurons. They compress, the flexors and the extensors compress and relax alternately, and that's what gives you movement. And the phase relationship between the neurons that drive these effectors is what gives you the different gaits that you see in animals, right? So so this was, of course, the gait transitions in animals. Now, it's not just movement, right? These oscillators can, of course, generate spatiotemporal patterns that are useful across the brain. Now, here's an example, and this is from, I think this is a paper from '96. I should have probably put this up here. This is from Gilles Laurent's lab. And what they've done here is, you see these faint gray lines out here. So these are recordings from the antennal lobe, which is the equivalent of the olfactory bulb, except in insects, it is the antennal lobe. And they're recording from neurons in the antennal lobe. So what they do is they present different odors to the antenna of insects, the equivalent of the nose. And depending on what odor you present, different neurons will fire at different times, right? So what is a rose in the patterns that are generated by these neurons? A rose in the space of the antennal lobe is essentially a selected group of neurons that fire at specific times. So it is a spatiotemporal pattern that's generated out here, right? So just like you have movement from oscillators, you can also have representations of pretty much anything using these connected networks of neurons, right? Yes. These are from single neurons. No, these are single neuron recordings. What you see are spikes out here. So they've done multiple, recorded from multiple neurons using the same order.
So understanding why optinol gives all these sort of intense grouping of spiking, whereas mint is
So at a network level, one can, of course, model this. So what's happening is that if you look at the antenna of the insect, there are these odor receptor neurons that are present. Now, when the odor comes and falls upon the odor receptor neurons, it leads to a cascade of activity that then goes to the antennal lobe. And it's a highly recurrent network of neurons. And so the odor-driven activity goes to this network, and it sort of starts driving the activity out there. So you will see many different patterns depending on which sub-network you're stimulating with the odor. So my sense is that it has a lot to do, the specific pattern that one sees has a lot to do with which neurons are stimulated, when it is stimulated, because there is sometimes a latency to that stimulation coming in, and the topology of the subnetwork. So there are both inhibitory neurons and excitatory neurons out here. And I know that the inhibitory interneurons in the locust antennal lobe, which is where this is taken from, have these conductances which give rise to adaptation, meaning they fire, but the time between the spikes sort of slowly decrease. And just as we had this flipping kind of alternating pattern, you'd get that in the interneurons, which in turn would also be reflected in neurons like this. But overall, I think it is a network phenomenon, and it's hard to a priori predict that this neuron would do this, whereas this would do something else, because it is a larger scale event. Though I should mention that activity like this, to the best of my knowledge, is quite unusual to have these alternating bursts. You typically have the projection neurons fire once or at some point of time and then quiet down, something like that. But this is one of the rare occasions, and I quite love this pattern of spikes. Mostly because it adheres to my favorite theories. Now you're going to go after innovative, yeah, ethanol. Hopefully, it doesn't get to be methanol. All right. So here, what you have is spatiotemporal patterns of activity using what are intrinsically oscillators. Connect them in a network, and you can get various patterns of activity. Can one sort of stretch this to look at other subcircuits in the brain? So let's look at the kind of spatiotemporal patterns that we're going to try and reproduce using our model framework, right? So the experimental setup is this. You essentially have some sort of a closed arena, and you're recording the activity of neurons, often in the hippocampal formation, in the entorhinal cortex. And you're also tracking the location of the animal as it moves, all right? So a lot of the images that you will see will look somewhat like this, right? So the black lines that you see out here, that's the path of the rat as it's going around. So I know that experimentalists seem to throw chocolate in the enclosure and have animals go around randomly. And here, this is most likely what's known as a place cell, and it's recorded from, I think, CA1 region of the hippocampus. So you have the animal going around in this rather random-looking trajectory, and the red dots here tell you the locations in space when a single neuron that you're recording from generates spikes. And you'll see that it spikes in a spatially circumscribed region. And when the rat exits that region, it stops spiking, right? And it doesn't matter which direction it's coming from, how fast it's getting here. None of that matters. Once it gets to this location, you have this particular neuron spiking. And if you look at another neuron, it probably spikes out here, another out here, and so on. This neuron spikes when it reaches the border of a region. Here, they've plotted the firing rate of a particular cell, and this cell fires when the animal is pointing in a particular direction. Right? So these are all different location-sensitive neurons. The one that I'll be focusing on is this remarkable cell called a grid cell, for obvious reasons. So here again, you have the path of the animal. And as it's going through the room, every time it lands up at one of these locations, it starts to fire. And when it leaves that region, it stops firing again. And if you look at where these regions are located in space, what they found is that it has this hexagonally symmetric grid-like pattern of activity. Right? So it's a rather remarkable crystalline structure that you see in the activity of neurons. And this is what we'll try to model using networks in the medial entorhinal cortex. All right? Okay. All right, so there's the hexagonally symmetric structure. And for work done, so John O'Keefe was the first person to discover these place cells. Edward and Maybrit Moser and their lab discovered the grid cells, and they won the Nobel Prize for this. I think it was in 2014. They got the Nobel Prize in Physiology and Medicine. All right. So this brings us to the task of how do you go about building a network of neurons that would generate a pattern of activity which has this really spectacularly symmetric pattern. And luckily, we are going to hear the Turing lecture soon, but one of the ways to arrive at these patterns turns out to be related to work done by Alan Turing. This is his 1952 paper called The Chemical Basis of Morphogenesis, where he talks about how you can start off with a rather amorphous substrate. He was interested in trying to figure out how patterns come up in development, right? How I or all of us have these elaborate faces and 5 digits and so on, because if you look at a sperm and an egg and you put it together, it's this rather amorphous jumble of chemicals, and you end up with the remarkable patterning, right? So Turing was wrestling with that particular question, and he came up with an interesting theory of how these patterns could emerge. And a great deal of this theory goes into the making of the grid cell patterns that we see in rats. So the first thing is, of course, to build a network that will generate these hexagonal patterns, right? So that's one. But these hexagonal patterns, it's not like a static pattern, right? The animal's running around, and as it runs around, you'll see that neurons start to spike, right? So this pattern is not just it's connected to the movement of the animals. How do you connect this particular hexagonal pattern to the movement? So these are the two things that we'll have to wrestle with in order to come up with a model. All right, so let's talk about some of the attractor models that exist and that some of us are probably quite familiar with. One, of course, is that you could start off with a very simple caricature of a neuron. You can think of it as an on-off switch. People have thought of it as spin-up, spin-down states, and so on. So if the neuron is on, it's 1. If it's off, it's 0. And you can think of it as these 0-1 switches. All right. So how do you build pattern into this? So there are two kinds of interaction that you can have between neurons. One kind of interaction is excitation. So if one neuron excites the other neuron, it has a tendency to drive that neuron to fire, right? If one neuron is on, it's going to drive the other neuron to be on. The other kind of connectivity that you can have between neurons is inhibition. Now, mind you, this is all a caricature of reality, but it's a caricature that's just designed to understand how things operate. But there are several caveats to everything I'm saying. All right, so inhibition means if one neuron fires, then it compels the other neuron or takes the neuron further away from threshold and will likely turn it off. Right? So these are the two kinds of interactions that one can have between these switches. Now, here's an example where you have a bunch of neurons. They're all-to-all excited, right? So each neuron is connected to all the other neurons in this array. And there are a bunch of neurons that are on, and there are a small number of neurons in there that are off, right? Those are the ones in red. And the connectivity is all to all. Let's say that the connection between them is excitatory. Then at the next point in time, you will likely have all of these arrows pointing in the same direction, which is up, because these neurons would drive the other neurons to fire. All right. Now, how can you start embedding patterns into this kind of a network, right? So one way to embed patterns into a network is to introduce inhibition. All right. So let's say you wanted to have these lines in this array. You wanted to have one column that was on, another that was off, a third that was on, and so on. One way to do that would be to have inhibition from all the neurons that are on to all the neurons that are off, right? And vice versa, right? You can have the off neurons connected to the on neurons. So if two neurons are going to be in different states, you want them to be in different states in your final pattern, you just have to connect them by inhibition. And if you want them to be in the same state, you connect them by excitation. And when you do that, however you perturb the network, it's going to go and fall into this kind of a state where you have alternating lines that are up and alternating lines that are down. All right. Now, if you wanted to make an associative memory out of something like this, which is what Hopfield did by trying to figure out what are the weights that one needs to have in order to embed a pattern like this. Well, you can embed a pattern like this by deciding what your excitatory and your inhibitory connections have to be. So all the gray blocks would be connected by excitation, all the white blocks would be connected by inhibition, and across the gray and white blocks, all the white blocks would also be connected by excitation, and across colors you'd have inhibition, and you could embed a pattern like this. Now, if you introduced noise out here, what you'll see is that you end up in you'll recover the pattern that you had put in. So what Hopfield did was, of course, figure out a way in which you could embed these patterns, figured out the constraints on a rotodynamical system that would give rise to a way in which you can embed memories into a network like this. And he, of course, got the Nobel Prize in Physics last year for this one. All right, so here you have attractor models and you can embed patterns in these. What about hexagonal patterns? Of course, you can embed hexagonal patterns too. So if you have an array of neurons, the ones in the middle, let's say these are on and these are off or inhibited, you could have all of these guys connected via excitation. The surround is connected by inhibition. And when you do that, you will get a hexagonal pattern of activity. And that's the kind of pattern that you see that's the kind of pattern that grid cells generate when they fire, right? Except what I'm talking about here is a sheet of neurons, all right? What you're looking at out here is a particular neuron firing at a given location. So there is this transformation that I'm making from real space, which is the physical space that we are walking through, to neural space. And can you actually transform one to the other? It turns out that there's a lot of evidence that this transformation is a reasonable assumption to have. So it does seem like the grid cell network is locally connected, it's topographically organized, and as the animal moves, not only do you see this kind of activity, you also see activity on this sheet moving in a manner that resembles the activity or resembles the physical movement of the animal. So there is a transformation between the real and the neural space, and we do have to figure out what the exact kind of transformation it is, and so on, right? So how does one typically build an attractor model? So first you need to have a way in which you generate these patterns. So local excitation, long-range inhibition, right? So that's our pattern of connection. So if you look at the connectivity kernel, it's going to have this kind of a form, symmetric form. And when you look at the firing rate, you're going to get a pattern of activity which is also spatially periodic. And this is in one dimension, right? Now, one can easily extend this to two dimensions. So if your connectivity kernel, and this is work from Burak and Fiete, your connectivity kernel has this central region, which is excitatory, and there is an inhibitory surround. So that's the kernel. If you were to take a slice through here, that would be the form of connectivity. And if you have this connectivity kernel, you will end up with a pattern of activity that is hexagonally symmetric. The scale of the activity depends on the scales that are present as far as the inhibitory networks, as far as these connections go, right? So this defines a scale in the network, the distance between the vertices in a network. Now, one could ask, why do you have a hexagonally symmetric pattern? Because you have radial symmetry in the connectivity, and that gives you hexagonal symmetry, because think of it as a way to pack the most number of circles into a region, right? And that's the kind of activity that you're doing. The length scale is actually determined by the connectivity of the. Are you asking about the model in the model? Yeah.
There are
Right.
distance between those values.
Right. Right. So we think, we don't know the complete answer, we think that it is related to the connectivity of the inhibitory network. So it is so if I were to take a particular inhibitory neuron, how far does it go out to influence other inhibitory neurons? It turns out that there's very little excitatory connections in.
Above the enclosure, the grid doesn't change.
The grid doesn't change, though you can change. So there are perturbations to the grid that come about when you say shear the grid. You will see changes that take place. And there are some theories that describe how that might happen. Some of it related to, well, how do you pin it to the boundary? There are some theories related to oscillator interference models that tell you how these shear can happen. But largely the assumption out here in this model is that the grid scale is related to the connectivity between the inhibitory interneurons. And there are other changes that can happen that are due to the intrinsic properties of the neurons, which we'll talk about.
Grid sizes of different, there are neurons with
grid sizes of different, yeah. So in fact, when you, so if you look at the entorhinal cortex and you go from the dorsal to the ventral extent of the entorhinal cortex, the distance between these vertices start to increase. Not only does the distance increase, the radius of these points will also increase as you go along the dorsal to the ventral axis. And what we also know is that this change is not a continuous change, but it changes in discrete jumps. And the first papers on the subject thought that it was something like about square root of 2 was the ratio, but it seems more likely that it is n by n plus 1. That's the geometric series that that it follows as you go along the dorsoventral axis. So yeah. Okay. So okay. So here's your real space versus neural space, and there does seem to be some mapping between real space and neural space. Now, the second thing, so the first thing, of course, is you want to make a hexagonal grid. And that turns out, thanks to Turing, is a rather straightforward thing to do. You need radially symmetric connectivity, and these patterns will arrange with a hexagonal symmetry. Now, what you also need to do is you need to couple the activity to the movement of the animal. Right? So when the animal moves, the activity pattern has to also change. Some other neuron has to fire and so on. So how do you do that? So one way to do that is that you can introduce an asymmetry out here. All right. So you can have some neurons that have an asymmetry. Let's say we are looking at this, let's say we are looking at a rightward movement. All right. So you can have some neurons which have an asymmetry that is just tilted to the right. You can have other neurons that have an asymmetry that's shifted to the left. Okay. And all of these neurons are, of course, randomly distributed on either a line or a sheet, depending on what your attractor model is. So if I were to stimulate only the sub-network that has the rightward asymmetry, it's going to move my attractor in one direction. If I choose the other symmetry, it's going to move it in another direction. And if I sort of have a mix of both, you can sort of like using a joystick, you can control how fast it goes in one direction or the other. So that's in one dimension. If you wanted to do the same thing in two dimensions, one way you could do it is that, let's say you have these 4 neurons that are picked on the sheet. Each of them has this radially symmetric inhibitory field, but you have an asymmetry that's introduced. You have an asymmetry that's introduced in one of the 4 cardinal directions, right? And you can use this to simulate the movement of the grid in one or the other direction, depending on which way the animal is moving, right? So you can imagine that if you have an electrode, so let's say that this is the neural sheet, all right? And you have an electrode out there. You'll see neurons fire. So the animal's moving rightward, and you'll see that these neurons start to fire every time the bump crosses that particular location of the electrode, right? So in this manner, you can actually couple the activity of the neurons to the movement of the animal. Now, here's the problem, I think, or here's an issue with models of this kind, right? So all of these models that I've looked at form a sort of class of models known as attractor models, and they're typically constructed using rather simple dynamical systems at the nodes of the network. So here, if you look at the network equations, this is the network equation that you have, right? So S can be thought of as either the activity of a rate-based neuron or the summed activity that a neuron is getting as input. So that's, if you look at the intrinsic dynamics of the system, it's just this linear thing, right? You perturb it and it's going to exponentially come down or go up depending on the numbers out here. And this is the connectivity, which is, in this particular case, it's a function that's flat, and beyond a threshold, it just rises linearly, right? So that's the kind of function and the bias. So that's the form of the neurons that one uses out here. Now, if you were to stick an electrode into the medial entorhinal cortex, you'll find that the neurons in the entorhinal cortex do stuff that is far more complicated than this really simple rate-based neuron does, right? So here are some of the things that you could get a stellate cell to do. So what's happening out here is that one is giving this neuron an inhibitory input at about this time, and at some other point in time, you release it from inhibition. And when you release it from inhibition, this neuron will tend to go and generate a spike, right? And depending on the strength of inhibition, the timing of that spike can be moved around. So this is called post-inhibitory rebound. And it's a really interesting property that you see in these cells. If you inhibit them, they show currents over different time scales, SAG currents, that are due to very specific channels that are present in grid cells. The sub-threshold properties of these cells are such that they also resonate and have very specific resonance frequencies. And as you go down the dorsal to the ventral side, the resonant frequencies change continuously as you go from one side to the other. And that's also determined in large part to what are the channels that are expressed in these neurons. So in a sense, grid cells, real grid cells, seem to be a far cry from the model grid cells. And the question is, have we reduced things too much? Do we need to increase the complexity? What does the complexity buy you? These are the kind of questions that we're struggling with. And Inayat started, Arun before him, and Inayat started working with somewhat detailed conductance-based neurons like this, trying to generate grids. And it turned out to be quite an interesting, sorry, 2 minutes? Okay, thank you. All right. So how does one go about, I get carried away, you should stop. So how does one go about making an attractor? Well, the first thing is that one needs to construct a switch in order to make an attractor, right? And how do you take these rather complex neurons and construct a switch? One of the ways to do that is you take two inhibitory interneurons, you connect the stellate cells like this, and you have this the stellate cells giving a projection to these inhibitory interneurons. Now, when you look at the medial entorhinal cortex, what we know is that there are very few stellate to stellate connections. There's very little lateral excitation between stellate cells, but there's a huge amount of recurrent inhibition, and the stellate cells talk to each other through these inhibitory intermediaries. So it turns out that this particular structure gives you the seed of what one needs. It gives you the ability to construct a switch. The next thing, of course, is to connect these switches to form networks. Here we have connected them into a circular network. It's basically a ring attractor, periodic boundary conditions. In the middle, you have inhibitory interneurons, and you have stellate cells on either side. We introduced an asymmetry by making this stellate cell not connect to the one directly, to the inhibitory interneuron directly below it, but out here, and this stellate cell connects out here, right? And so by introducing this asymmetry, depending on which of these rings you stimulate, you can drive the neurons to go in one direction or the other. You can change the sizes of the grid fields by changing the connectivity, the distance between these peaks of the inhibitory connections. Now, we were also able to move this into 2 dimensions because, after all, you have a 2-dimensional attractor. And the connectivity scheme is largely the same. You have to have these asymmetries, directional asymmetries, and it's periodic on a plane, so you have tori, and this is the asymmetry you have. You have a torus consisting of stellate cells. It connects not to this inhibitory neuron, but to something that's slightly shifted with respect to it. And when you do something like this, you can actually generate patterns of activity that resemble what you see in, so here's the movement of the animal, and here are the different sheets, each of which stands for east, west, north, and south. And you'll see that the activity of neurons closely follows the trajectory of the animal as it navigates space. Right? Okay. Now, the question, of course, is why build a complex model when a simple model? Okay. I also had a number of jokes written out here, but I'm going to skip through those. All right. So key thing, of course, is that why make these complex models? It turns out that by making models of this kind, we are able to construct representations that predict where the animal will be. So you can infer the location of the animal by looking at the activity of neurons. And if you look at a rat running along a track and you look at the activity, it doesn't tell you necessarily about where the animal is located right now. It tells you where the animal is going to be located a little while later. So that's the predictive representation that you see in experiments. And what we found is that this particular predictive representation was easy to generate, provided we installed the right conductances into these neurons. When you have these hyperpolarization dependent conductances that are present, we found that we could, in fact, have a predictive representation that picked the location of the animal that was about 5% the length of a grid. And the prediction, of course, being that if we knock this particular channel out, that predictive representation would fall apart. I think I'll stop here and maybe take questions. I think I'm over time by a bit. Yes.
Time delay, Collins?
In this, so we don't have time delays in this. We not explicitly put time delays in this. The times that come up out here are basically once you stimulate a neuron, there is a time scale for the synapse to. No, there are, yeah. Yes.
Generate a certain, complexity by having a small time delay.
It's yeah. So I guess one of the
Signals that travel.
right. They are signals that travel by at finite speeds, but I think one should also think of what are the timescales of the system that are important. So here I would think that there are two oscillations that I would use to think about the clock of the system. One is theta, which is of the order of 10 hertz, and the other is maybe gamma and high gamma, which would be of the order of anywhere between 40 to even close to 100 hertz for fast gamma. And within those time scales, and given the spatial extent, I'm not sure what percentage would be affected by delay. So a theta oscillation, quite unlikely. It would be a very small percent. But I think with gamma and fast gamma, time delays would play a role. But I think the primary reason I'm not using time delays is because they're just very hard to wrap your head around, right? Because it's an infinite dimensional system straight off. Yeah. Right.
So that you can generate sort of,
Right.
hugely complicated
Right.
patterns.
So these are sort of axonal conduction delays that one would have to think about, and the spatial extent that one is looking at is of the order of centimeters, and the propagation speed is yeah. Right. So we don't have it in here, and one would have to check whether it will be important. Yeah, I'm a bit scared of time delays, and that's the primary reason. Visceral discomfort. Yeah. For the
For the
oh, these are not grid field sizes. So one is.
But you do get such variation in
Yes,
some
you
axis
do,
of the
depending on
hippocampus,
what depending on the length scales that you use.
right? So in the model, what makes this difference? Is there a difference in the conductivity or how much of the IH channel?
No, so the difference in the scale is due to the extent of the inhibitory connectivity.
I see. So
Right?
that's the only.
That's
parameter
what
that you
gives.
take.
Yeah.
And is there evidence for that in the?
There is some evidence for that because it is topographically organized. There is evidence. Actually, there is more evidence for changes in HCN, but there is certainly some evidence. Yeah, there is certainly some evidence for how much. Well, not so much, but there is some evidence for the spatial connectivity changing over the dorsoventral extent. The HCN also has an effect on both the grid scale and the time, but it's a much smaller effect than one has due to the connectivity of the network.
Okay, and in your model, if you were to change the HCN.
So what the HCN does is it advances the onset of the next spike. So in our case, if you're looking at layer 2 stellate cells, then the layer 2 grid cells, what you see is that the prediction extends to about 5% of the length of the diameter of a grid field. And in order to get that much prediction, HCN channels are sufficient. Right? So just having HCN would give you that, which also gives rise to the prediction that as you go along the dorsoventral extent, the extent of prediction would also change. And that's something you do see in experiments. The one thing people haven't checked is what would happen with knockout. And our prediction is that you should lose that 5% predictive part in knockout. If you go to layer 3, there, the prediction of the representation is about 25% of the size of a grid field, and you wouldn't get that from HCN. So there you need to have an asymmetry in the network kernel, which I didn't have a chance to talk about. So you'd have to have this kind of an asymmetry in the network kernel, and that would give you this kind of a thing. So in layer 3, it's pyramidal neurons that are grid cells, and they don't express HCN like stellate cells. So this seems to be a more likely candidate. Yeah. All right. Thank you.